My name is Leanna, and I want you to see me and see an F.
My name is Blake, and I want you to see me and see an F.
My name is Jayshawn. I want you to see me and see an F.
People who have NF are so varied in different types.
I basically tell them that it's genetic disorder, can cause tumors to grow anywhere on or in the body, and it's like something that you're born with.
I look healthy on the outside, but on the inside I have over 100 tumors in my body.
But what people don't see with my diagnosis is that I'm losing my hearing.
They just see this person, oh, you're really in pain every day?
I have NF1. I'm the only person in my family that has NF.
They make assumptions that they're not just like every other teenager.
Because some people are different than other people.
I see you as someone who's different, but actually understands you and knows a part of understanding of what you're going through.
All of my tumors are internal. I don't have any skin tumors.
One of the things people can't see about NF is the constant worry we have, knowing that a tumor could be growing anywhere in their body at any time.
I'm not too sure. I might develop tumors, I might not.
I think about the future and I get scared, but then I meet people within CTF and the NF community, and it's inspiring.
Just because I have NF2 and I've had 27 surgeries, despite these challenges, I'm still pursuing my dreams.
Someone could show a scientist, and then they could figure out a cure and keep on experimenting.
Although I had NF and I went through my own struggles, the second I found out my son had it, my entire world changed. And it was my mission from then forward to find a cure or an effective treatment.
The more attention there is to it, the more research, the greater chance they're going to find a cure.
We want everyone to support her and support everyone dealing with NF in the way we've felt so supported by the CTF community and the NF community.
My name is Jesse. I want you to see me and see NF.
My name is CJ. I want you to see me and see an F.
Please welcome Cara Anstead and Miranda McManus.
Somebody save your soul, 'cause you've been sinning in the city. I know too many troubles, all these lovers got you losing control. You like a drug to me, a luxury.
Hi everyone. If you guys could advance this one more slide to start. For anyone that is just walking in, we have a little pre-presentation survey that we want you guys to complete. I think most people have already gotten that done from what it appears, so we're going to move on pretty quickly from here. But if you do have technological difficulties with completing this survey at the beginning, there are some folks from CTF that are walking around that have paper copies, and they can give you one of those to help you out. All right, so I'm Miranda McManus. I am a parent of a nearly 18-year-old son with NF1. I'm also a scientist, and I teach biology at the College of Charleston in Charleston, South Carolina. This is Cara Anstead. She is a genetic counselor at NYU Langone Health in the Comprehensive Neurofibromatosis Center. And we're speaking here today on behalf of the RANGE Gene Therapy Group because gene therapy is something that is on the horizon. There are gene therapies that are approved for a number of conditions right now. We're still a ways away in NF, but we need to get prepared because there's a pretty steep learning curve to understanding gene therapy. And so we want you guys to help us understand how we can best prepare the community to get ready for gene therapy to get started, right? So that's what this talk is about. And so we're going to start off with some basics of genetics, and then we're going to move on later and talk about different types of gene therapy and the status of gene therapy in NF. And because this is a pretty complicated topic, we're going to have breaks along the way. And so as we get to those breaks, we're going to give a chance for questions. For these short breaks in between, I'm going to ask you guys to keep the questions topical to that part of the presentation. And then we're going to open it up for broader questions, as well as have a little discussion at the end, all right? So when we talk about gene therapy, it's all related to genes, right? And if you notice on our title slide, we're actually calling it gene-targeted therapy. And the reason for that is this is a process going from DNA, which kind of holds the instructions for everything that happens in our body, to proteins that do the work. And so when we're talking about gene-targeted therapy, it can impact any part of this process, all right? So this is also known as gene therapy. It's also known as gene-directed therapy. And Kara and I are probably going to switch back and forth a little bit between those terms as we go through. But they're all the same thing, all right? Really, the only reason we're using gene-targeted therapy is so that you understand it's not just the genes that are affected. It can be at any point in this process. So we want to tell you a little bit about how genes do the work in our bodies. So genes are little segments of DNA. So you know the DNA is like our instruction manual for everything that happens. And genes are just little pieces that code for specific proteins that do work within our body. And this process goes through a middleman called RNA. So if you notice at the top up here, we have our gene. And this gene segment, you notice, is separated into different parts. So there's exons, and there's also introns. So exons are the pieces that actually code for that protein down at the bottom. And along this process, those introns end up getting chopped out, all right? But just like everything in our bodies, it's a little complicated the way that it happens. So we start out with the gene, and then it goes through this process called transcription. Transcription is when an RNA molecule is made as a copy of this gene, and it's essentially in the same language. DNA has a language of letters, right? And RNA does too, and pretty much the same letters. And then after that initial transcription step, there's an extra processing step that happens to where all those intron pieces end up getting chopped out, all right? And then we get this messenger RNA molecule. So most of you have probably heard of messenger RNA because of the COVID vaccines a couple years ago. Most people hadn't heard of it. So messenger RNA is essentially just a message that takes that DNA code and takes it out to the machinery that ends up making the protein. So that next step is what we call translation. And in that translation step, the RNA is translated into a different language. So this is the language of amino acids that ends up making this chain of amino acids that we call a protein. And there's some steps that happen there too, because you notice this protein at the bottom is really folded up. And if that protein isn't folded in just this right way, then it doesn't have the right function, right? So when we're talking about gene-targeted therapy, we're talking about what happens anytime along this process. And just so you know, when we're talking about NF, the proteins that are important in NF have some pretty major roles in our body. So for one, they can prevent cells from over-multiplying, right? And they also play a role in nerve cell signaling. And so the way that signals are passed around your body and the way different parts of our body communicate with one another. So when somebody has NF, they have a type of variation in that genetic code within the genes that code for, and we're talking about NF and schwannomatosis, right? So when I say NF, I'm kind of talking about all of these different conditions that we know exist. And so there's these genetic variants that occur, and there's different types of variants that happen. And when there's what we call a pathogenic variant, or you may have heard of it as a mutation, then that ends up causing the condition of NF, right? So there's different ways that this can happen. So a good analogy for this is just using a sentence, right? Because remember that DNA code is made up of letters. And so sometimes one of those letters might change. And so if our original sequence was something like the gray cat ran down the hall, then we might have a missense mutation where one of those letters is changed. Then it might change to the gray cat ran down the ball. Or we could have an insertion mutation where a piece is added. And that piece can just be a couple letters, or it can actually be thousands of letters. And sometimes it's a whole new piece, and sometimes it's a copy, so we end up having a duplication happen. So we might end up having something like the big gray cat ran down the hall, or the gray cat ran down the hall, right? We can also have deletions. So sometimes a chunk is taken out. And so we might get something like the gray ran down the hall. And again, this can just be a small piece, or it can be a really long segment of DNA that ends up getting chopped out. And then we can also have what we call nonsense mutations. And a nonsense mutation is when the sequence gets cut short. So that period gets stuck a little bit too early in the sentence. So rather than having the gray cat ran down the hall, it might just stop at the gray. Can we move forward to the next slide? Okay, so I'm gonna pause right there because that's a lot of information, right? And give you guys a chance to ask any questions because I don't want anybody to get behind. So the CTF folks have mics. Does anybody have any questions about this process so far?
Oh.
Yeah, don't be shy.
I see one in the
We want to make sure that everybody understands this as we go through. Yeah. Here, wait for the microphone so everybody can hear you.
Hi, my name's John Duder. Are all those types of mutation, if that's what you call them, equally likely, or are some more probable than others?
Yeah, so, some are definitely more probable than others, but they're all pretty common, right? And one really complicated thing about NF and all the NF-associated conditions is that there's a lot of different types of mutations, right? We're not just talking about a single mutation, like with some conditions that might happen. There's a whole lot of different mutations that can occur. Yeah.
Yeah. Hi, I'm Camille.
Yeah.
With all those, have they noticed that all of those can happen when NF and might be related to the severity of different?
Yeah, so they definitely could. So we know that in cases where the whole NF gene is deleted, that there's going to be a more severe, what we call phenotype, which is the way that that shows up in a person, right? But unfortunately, it doesn't even seem that the same mutation causes the same problem in families, right? And so that's a hard question to answer. We don't actually really know, and it would be nice to understand a little bit better how these different mutations do cause NF.
And most of the time in NF1, the type of mutation you have does not predict severity or the types of symptoms. It's a little bit different for NF2, so there are some associations within NF2-related schwannomatosis, so having a nonsense variant or something else that cuts off the gene is more likely to cause younger onset NF2 than if you have a missense variant. So it depends on the type of NF that we're talking about.
Absolutely.
Yeah. We'll take one other quick question.
What was the second major role of NF-related proteins that was to prevent certain cells from multiplying too much? What was the second one?
So it can have an effect on nerve cell signaling. So, our nerves pass around messages in our body, right? And so when there is an NF variant, sometimes that can be impacted.
Yeah. Especially when we think about like the learning impacts of NF1 and how our brain cells communicate with each other.
All right. So now we're going to talk about a little bit more specifically what happens in NF, right? So we have many genes in our body, but when we have a mutation in the NF gene, that's what causes NF. And in most cases, this is going to be a mutation that shows up in every cell of your body, except for in the case of mosaic NF, for example, right? And that shows up before birth. So this is, your genes are passed down from your parents, but so sometimes that NF mutation can be passed down, or sometimes genes just mutate. Sometimes these variations just arise, right? And if you have NF, then before birth, you notice that little X popped up on the screen here. This represents that pathogenic variant, and it's only going to be on one copy of your DNA. So we each get our DNA from our mom and our dad, right? So we end up having 2 copies of every single gene. And if you're born with NF or schwannomatosis, then you end up just having 1 copy that has this variant, right? And so in that process of transcription, which remember is when this middleman molecule, this intermediate molecule RNA is made, that means that that variant is also going to show up in the RNA, right? And that ends up impacting the way that the protein is made. So sometimes you might not get any protein made at all, or sometimes it might impact the folding of that protein molecule. And if you remember what I said before, that folding is pretty important for function. So it doesn't work properly if it's not folded properly. And so that ends up rendering 1 copy of NF essentially non-functional, right? Now, the good thing is you still have 1 copy, and that 1 copy is still functional, but we kind of need 2. And so if you don't have that second copy that is functional, that's going to lead to some of the symptoms of NF, like developmental issues or learning disabilities, scoliosis, vascular problems in NF1 and NF2 or schwannomatosis. This also leads to issues like cataracts or chronic pain, particularly in schwannomatosis. And so there's a lot of different types of issues, symptoms that can show up just because of missing this 1 copy. And again, there's a lot of variation there, right? So sometimes people may have much less severe symptoms than others, and that's really going to be down to how well that other copy is working, or a whole lot of other factors that we don't fully understand yet, right? So I'm going to pause again. Any questions? So we have one over here.
Oh, wait.
Can you wait for the microphone? She's running.
Hi, I'm Laura Colderette. My daughter has NF2, yet she also has learning disabilities, scoliosis, which you have under NF1. Do they share some? I know one is NF2's 22nd chromosome, the other one's 19th. Can you have both, or can you share symptoms between the 2?
Yeah, you want to take that?
So generally we don't see differences in learning and scoliosis associated with NF2 related schwannomatosis, and we don't see overlap between these conditions. They're separate genes in separate parts of our DNA. It might be possible for your child that there's other things that are playing a role in the symptoms that she has, but I wouldn't expect that it's NF1 related. I'm not aware of any cases of learning differences in people with NF2 specifically.
Yeah. And as most of you probably know, it can be very difficult sometimes to differentiate whether a problem is NF related or whether it's just, normal kid related, right? Because there's a lot of differences between each of us as it is.
Yeah.
Anyone else?
Hi, my name's Melanie and I have NF1. Based off her question, I have hearing issues, and I didn't know if that was related to NF1 or NF2 specifically, because I heard it's more NF2 side instead of NF1. So I didn't know if that's related or not.
That's a good question. So the cause of hearing issues in NF2 related schwannomatosis is from the growth of tumors on the hearing nerves. There are some people that can have hearing differences in NF1 due to a different cause. So some people have what's called NF1 Noonan type or Noonan syndrome type, where you have a little bit of overlap with another developmental condition called Noonan syndrome, which can cause things like heart defects or a little bit of minor hearing loss, but from a different mechanism than the tumor development that we can see in NF2. In general, most people with NF1 don't have hearing differences, but there definitely are some people that do. Yeah.
Anyone else?
Maybe I see one at the back, maybe. There.
Can you explain mosaic NF, please?
Yeah, so the mutations that happen that cause NF happen early on in development. So sometimes it's right off the bat when there's just a couple cells there, and sometimes it's later on. And so, you know, through our development process, the cells of our body divide, right? And so if it happens later, then you can potentially have NF in certain segments of your body, but not in the rest, right? And you're still gonna have some symptoms of NF, but if they were to do a genetic test, for example, in certain ways, it might not show up because in some of your cells, you don't have NF, right?
And you can only be mosaic for neurofibromatosis if you're a sporadic case. So if you inherited NF or schwannomatosis from your parent, then it was in the egg or the sperm cells, so it's in all of the cells of your body. But depending on if you have NF1 or NF2 or the other types of schwannomatosis and you're sporadic, there's different rates of mosaicism. So mosaicism is very common in schwannomatosis, it's less common in NF1. In the interest of time, I think we're going to move on to the next section, but we can take more general questions, and everyone's welcome to come up and speak to us after the session as well, because we'll be having a short break.
Absolutely. So for this next part, I'm going to hand this off to Kara.
Thank you very much. So we talked about the genetic changes that are present from birth in NF and schwannomatosis and cause certain types of symptoms, but when we think about NF and schwannomatosis, we primarily think about the tumors that can develop in different parts of the body. And so I'm going to walk through what happens in our DNA and through the genetic process that leads to these developing. We've added an extra figure here on the left, so it looks exactly the same, and I'm going to walk you through what happens. So in any one cell, we're always being exposed to things that can mutate our DNA. We cause these somatic mutations. They happen in our cells that cannot be passed on to our children, so like our skin cells or our nerve cells or other parts of our body. So we are always being exposed to things that mutate our DNA. And what can happen is in one cell, that mutation happens to happen in the other copy of the NF gene. So if you have NF1 or NF2, then it's in the other copy of NF1 or NF2, and you can see that now that DNA has the mutation in both of the genes. For the other types of schwannomatosis, there's a couple additional mutations that happen along the same 22nd chromosome, but it's similar that you end up with no functional genetic gene copies of this. And we see that the mutation, it moves through to the RNA level. Did I click it hard enough? There we go. So it travels through the RNA level, so now all of the RNA has a mutation in it. You can see also that the X's are in different places and different colors because these mutations are different from each other. They happened at different points. And then it travels through and it impacts the protein as well. So maybe we don't have any protein being made or it's misfolded. And then that one cell now has no functional NF protein, okay? And that, as Miranda said earlier, that protein's really important for controlling that cell and how quickly it divides. Our cells need to divide a little bit. We make new skin cells and new nerve cells, but if they divide too quickly and too often, they become a tumor. And so what this leads to is the different benign tumors that we can see in neurofibromatosis, because that one cell is now missing this control on its cell division, and it multiplies and multiplies and multiplies. And so then you see things like a neurofibroma developing, or a schwannoma if you have NF2 or schwannomatosis, or a meningioma, or an optic pathway glioma. So that's what happens if it's in a nerve-related cell. If it's in your skin cells, particularly the parts of our skin that have our pigment in them, then that's what causes the cafe au lait spots and the freckling of NF1. Okay, so if you were to like take a biopsy of a cafe au lait spot or of a tumor and do genetic testing on it, you would find 2 mutations in each of those samples, okay, because it's missing the functional DNA. And in some cases, particularly in NF1, there's a risk of cancerous tumors that can develop. And usually that happens because then that tumor gets more mutations in other genes that are really important in controlling cell division even more. And so I've listed a couple of them here that are common ones that we can see. If, for example, a plexiform neurofibroma becomes cancerous, or an MPNST, or if you have a high-grade glioma from NF1, okay? These are all somatic mutations that happen through our lifetime that are only in the cells of that individual and cannot be passed on. Are there any questions about this section of the presentation? About what happens on the genetic level that leads to tumors developing in NF? See one over there.
So on the last point of your slide, when there's a transformation in the plexiform fibroma, and then you have like a more mutation that causes the cancerous to actually appear?
Yep.
Does that develop over time, or is it spontaneous as well?
Yeah, so this develops over the time of someone's life. And so you start with the one mutation that you were born with, right? And then you get the second mutation that causes the plexiform neurofibroma. And then some plexiforms, not many, only a small number of them, over time develop additional mutations within the cells of the tumor that cause it to grow uncontrollably and become cancerous. Yes.
So I think, and I may not have heard you right, but I thought I heard you say something like some kind of an outside force can cause that cell to mutate. So I'm a first-generation NF person. Both of my parents are what's called double downwinders, meaning they got basically contaminated by nuclear fallout from 2 different plants. Also, my mom was on a dairy farm with multiple different chemicals that are no longer in use. Now, could those, and I was in utero while she was on that dairy farm at one point, could that have caused the mutation to happen?
That's a good question. There's a lot that we don't understand about what causes mutations in our DNA. We do know that the process of copying DNA to make a sperm cell or an egg cell or to copy our own cells during our lifetime in our body is an imperfect process. I like to imagine it like if I was given an assignment to copy an entire book myself without spell check, I would probably get most of the copying correct. But I might make a couple mistakes along the way, and I'm able to do my own spell check and usually find them, but sometimes I might miss one. And the same thing happens through the process of our DNA being copied. We have different things within our cells that double check to make sure that everything is copied correctly, but sometimes it misses something. There is something about the NF1 gene. It's one of the largest genes in our body, so it's many thousands of letters that need to be copied during the process of someone being produced. And it's also maybe something about that part of the DNA is a little bit more fragile. We do know that things like if you had like CAT scans or radiation therapy, we know that mutates our DNA. Other things in the environment, I think we don't have as good of an understanding of what happens. But just general existence causes mutations within our DNA all of the time. The sun in the sky radiates upon us and causes mutations. So it's our cells and our bodies are really good at a lot of things, but they're not perfect because humans are not perfect. But yeah, we can talk more about that after, about your own experience. Were there any other questions about this piece of the talk? Maybe one more at the front.
Sorry, on the slide before this, you had tumors non-malignant, and one of them was meningioma. Now, does that play any role in NF, or was that just listed as a non-malignant tumor you could have?
So meningiomas are associated with NF2 primarily, and some people with SMARCB1 related schwannomatosis as well. Yeah. So that's a type of benign tumor that's very common in NF2-related schwannomatosis.
And just to give you one more piece of terminology that we haven't mentioned yet. So we talked about these changes that occur before birth, and then the ones that happen later, those somatic changes. When we're talking about the changes that occur before birth, those are germline variants, all right? You'll hear that term thrown around a little bit with gene therapy, so I want to make sure that you guys understand the difference there. So the germline variants are the ones that are there before birth, and somatic mutations or somatic variants are what occur later on in life.
And germline variants are what can be passed on to your children.
Right.
So I know you guys know that the title of this presentation is gene-directed therapy, and we haven't gotten there yet. So this is the slide where we talk about what is gene-directed therapy and what are we talking about different things that could be on the horizon for NF-related disorders and what they are acting on when we talk about the gene-directed therapy. So there are different steps in the process, and there's different steps where we have therapies in development. So one is gene replacement therapy. So that's using a virus to deliver a copy of the NF gene that does not have a mutation into certain cells of your body. So then you have like a backup copy that is functioning, making functional RNA, making functional protein. We also have, on the DNA level, gene editing, or CRISPR as it's commonly referred to, in which there's been a lot of mentions of CRISPR in the media. So what CRISPR does is you can see when that animation came up, the X disappeared on that copy of the gene because gene editing is making a custom CRISPR molecule that fixes your specific mutation, so it's no longer there. So then you have two functional copies of the DNA in the cell. So we can have gene therapy that acts directly on the genes itself, which is usually permanent because it's making a change to our DNA or adding an extra piece of DNA to our cells. But we can also act in different parts of the process. So we can have gene therapy that acts on the RNA level. So we can have RNA editing that specifically fixes a mutation on the RNA, or we can have exon skipping. If you remember, exons are different segments of the gene, so sometimes we can skip over the part of the gene that has the mutation, and then we have the rest of the RNA is functional. Because RNA is constantly being made and broken down, any type of gene-directed therapy that acts on the RNA is going to be temporary because it's going to be washed out as the RNA is constantly being replaced in the body. We can also have something work on the protein level. So sometimes we can make an artificial version of the NF protein, and then we can deliver it into the body, and so then you have more working protein in those cells. Or we can have something called nonsense suppression. And what that does is it acts on proteins that have nonsense variants, a very specific type of DNA variant impacting them. And it prevents them from being broken down so they still can work in some way. Just like with the RNA level types of targeted therapies, protein is constantly being made and broken down in our bodies. So any type of therapy that's targeting the protein is going to be temporary as well. Okay, so it does not permanently change your body. We can also have something called suicide gene therapy that's used to target tumors. It's in development in the cancer space. And what it does is it's a type of therapy that recognizes a tumor based off of its genetics or other markers on the tumor, targets those tumor cells and causes them to apoptose or commit suicide and break down on their own so that they stop developing and stop continually develop as a tumor. So these are different things that we could potentially use in the NF space from a gene-directed therapy perspective. There are pros and cons to all of these. So as I said, the ones that act directly on our genes are permanent changes versus ones that act on our RNA or our protein are temporary changes. Some of them can be done regardless of what type of mutation you have. For example, gene replacement therapy. It doesn't matter what type of variant is in your gene. We could all just give you a working copy of the gene. Versus some of them are very specific. So CRISPR is unique to every mutation. RNA editing and exon skipping is unique to that mutation or unique to where that mutation is in your gene. And nonsense suppression only works for certain types of variants. So it's not a one-size-fits-all solution for the different types of NF. There's also things that can be challenging depending on what type of NF we're talking about. So the NF1 gene, as I said, is very large. It's challenging to put it into a virus for gene replacement therapy. It's a little bit too big to fit. Versus NF2 is a lot smaller, so it might be more amenable to that type of treatment. So it might mean that we're talking about different types of gene-directed therapies depending on what type of variant you have in your gene, depending on what type of NF or schwannomatosis that you have. And so where are we with gene-directed therapy? So we have a long way to go in the NF space, but for other diseases, other genetic conditions, there are gene-directed therapies that are clinically approved and available for people that have these diagnoses. There are other ones that are in development. So gene replacement therapy is clinically available for some genetic conditions, including spinal muscular atrophy or SMA. Exon skipping and nonsense suppression are available for Duchenne muscular dystrophy. Protein replacement therapy is used for a lot of enzyme deficiencies, including Gaucher disease and Pompe disease. The other ones like CRISPR is not clinically available for any genetic conditions at this time. It is only in the research phase, but it is in human clinical trials for sickle cell disease right now. And the suicide gene therapy, they are looking at different research in the cancer treatment space, which we know from other clinical developments that have happened for treatments for NF. There's a lot of overlap between the types of treatments we use for NF and the types of treatments we use for cancer syndromes. So where are we with NF and schwannomatosis this year? So we do have some of these therapies are in development in animal models like mice. So gene replacement therapy is being studied in mice right now. So is nonsense suppression. We have others that are being studied in cell cultures, so growing cells in a lab and giving them the therapy and seeing what impact it has on those cells. And we're doing that right now for exon skipping, for nonsense suppression, and I think it's just those two, yes, those two. Protein replacement, we're not in a clinical stage right now. The NF proteins are really hard to make artificially and work in a lab, so we don't have ongoing research in that space right now. We don't have ongoing research right now for CRISPR and gene editing for NF, and we're not looking at suicide gene therapy specifically for NF at this time. Okay? There is a lot of interest, though, in the community with our researchers to try to develop different types of options of gene-directed therapies for NF. So I'm sure that this is going to be different in 2024 and 2025. Any questions about these gene-directed therapy options? I see a hand that went up very quickly at the front there.
Hi, my name is Sarah. I have a son with NF1 and he has a microdeletion, so he's missing the NF gene and a few others. And so we were really excited when we heard about CRISPR, and we've just kind of been waiting for anybody to be able to talk to about it. But it sounds like you're saying that the protein, like that section is just so big that it's like not stable enough to like recreate it in the lab. And then, but I was just so curious if you could just talk about how that even gets inserted into the body and how that would even affect somebody who's already developed and they're who they are. So how do you change that at a cellular level like that?
Yeah, I think the next section we're talking a bit about like how this is given and the impact it has, but there are challenges in NF in that some things develop very early. Certain symptoms develop very early, like you can be born with certain symptoms of NF. You could be born with a heart defect, you could be born with a bone abnormality, you could be born with a tumor. And when we think about also early development and the cognitive effects of NF1, that happens to different degrees through early childhood and even before you're born. So NF1 can impact even how our brain forms in utero. So there are limitations of gene-directed therapy. When we talk about what are going to be the goals of gene-directed therapy, most likely our first goal is to slow down the change of symptoms over time or to stabilize someone and stop something from progressing further. I think it's going to be very challenging for us to reverse symptoms that have already developed. So there are limitations to all of these therapies. And for microdeletion and large deletions of the gene, I think it could be challenging for us to figure out a way to give you back that large piece of DNA that is missing from your genes. Yeah. Other questions about this? I know we're running very long on time. I want to get our last section in. Okay. And then we can do general questions at the end, or please come up and talk to us if you have any questions. We're happy to keep talking through the break after this is over. So I just want to finish with how gene-targeted therapy might be given. Based off of what we know about where we are with the research now and the clinically available gene-targeted therapies. So in terms of timing, if it's a type of gene-directed therapy that's going to cause a permanent change in your DNA, that's most likely going to be a single dose. You get it once, it changes the genes in your body, and then you don't have to give it again, because then that DNA is constantly making functional RNA, functional protein in your body. If the gene-directed therapy is working on the RNA or the protein, as I said, that gets broken down. And so it's something that you're going to have to be given continuously, like other types of medications that you have to take continuously for them to keep working. So it might be something that you have to take every week, or every 2 weeks, or every month, or sometimes even every day. And when we think about how that treatment is given to you, many of us are familiar with IV treatments like chemotherapies. We call those systemic therapies. So then the medication or the gene therapy is given through an IV in the arm, travels through your bloodstream, and then hopefully is at a high enough dose that it can spread through your body and get to all the cells that we're trying to treat. That can be challenging, though, to get a dose high enough that it's able to spread through your body without causing a lot of side effects. So sometimes we do instead local treatments or targeted treatments so that you get the medication or the gene therapy a lot closer to what you're trying to treat. And in NF, that could be something that gets injected directly into a tumor. So for example, if that therapy is being used on a plexiform neurofibroma, it could be injected directly into it, so you could get a really high dose in that tumor. It could be that if we're looking at treating things like brain tumors or cognition, that it's a treatment that goes directly into the brain. So there are some genetic conditions where the gene-targeted therapy is injected directly into the brain so it can spread through those nerve cells very effectively. It's also possible that we might have gene-directed therapies that are injected through the spine, similar to like having a spinal tap, but instead inserting the medication that way so it can spread through your spine and into your brain. Again, probably something that would be considered for spine tumors or for brain tumors or for cognition. So we could have systemic therapies like an IV, but we could also have targeted therapies. And that could depend on what are we trying to treat, what type of NF are we trying to treat, and what type of gene-targeted therapy is this. And then I mentioned earlier with our question about what do we think are probably going to be the goals of treatment. Initially, it's probably going to be, can we slow down the change of symptoms? Can we slow down the growth of these tumors? Can we slow down other changes over time? We could also look at potentially stabilizing someone and halting their symptoms from getting worse over time. And then hopefully we get to a place where we prevent symptoms from developing, that this is something that you get given to you when you're younger to prevent things to develop as you get older. In general, there are some things that are really hard to reverse. So I think it'll be very tough for us to reverse symptoms of NF, but it really depends on what we're talking about and what specific parts of NF we're talking about. I think we're a long way away for this being a cure for NF, but I think there's a lot of promise for when we get to clinical trials that this is something that rather than just treating the symptoms, this is treating the root cause of what causes the different symptoms of NF. I know some of you are probably wondering, what are the risks? What are the side effects? Will this work? We don't know the answers to that right now. So it's really hard for us to know exactly what that's going to mean in terms of comparing gene-targeted therapies and looking at what is the best fit for you. But hopefully with more research, we'll have that information over time. And many times the very first clinical trials we do, we don't know if it's going to work, and we don't know what the side effects are going to be, which makes it really challenging. But we hope that this presentation has helped shed some light on what are we talking about when we talk about gene therapy, what exactly it is, and how it might be something that's available for our community in the next few years, so that eventually we do get to that point where we do end NF. Okay. Any questions about this piece of the presentation? I see a question there. Our microphone people?
I saw that one of them said for the intro to Brain 1, also for cognition. So would that mean things like ADHD and learning disabilities could be affected?
Yeah, definitely. Yeah. There are other genetic conditions that cause different effects on cognition, and sometimes that's the primary, primary symptom in those genetic conditions, and their gene-targeted therapies are being directed directly to the brain to treat those things. Because our brain has something called the blood-brain barrier, which is a really effective mechanism to protect our brain from things getting into it, but sometimes it makes it challenging for us to get medication into that space. So sometimes that's why we do medication directly into the brain that way. I want to open up if anyone has any other questions about the presentation, thoughts about the level of detail that we went into, or what you found really effective or ineffective. We really want to learn from you about what type of educational needs our community has, so that when we get to clinical trials, you have information that's helpful to you when you're making decisions about whether you think this is something that you want for yourself or for your child. So any comments or questions that anyone has about this? I see someone up here.
This will have to be the last one.
Okay.
Yeah.
Lydia, right
My question is, you talk about CAT scans, PET scans, MRIs affecting tumors, but so many of us have to have those consistently, so it's kind of like a give and take, or?
Yeah. So MRIs are magnets, so they don't have radiation in them, and most people, the primary scan that you're getting is an MRI to monitor your tumors. But it's true, PET scans, CT scans can have radiation, but we always have to weigh the risk-benefit of what that imaging is being used for. Sometimes it's really important to get that radiation in order to get the pictures that are important for your doctors to monitor you. Definitely.
And I wish we had more time, but unfortunately this is something that while we could talk about it all day, they didn't give us time to talk about it all day because there's so many other important things that you need to hear about at the summit. But we're going to be around. Please find us and ask questions if you have more. We would love to talk to you. The whole reason that we're doing this is we want to make sure that we're serving this population the best and getting prepared for gene therapy. Also, if you guys could advance the slide one more time.
There you go.
Here's that same QR code again. You may still have it open on your phone or whatever. And we have a post survey. It's basically the same questions that you answered before. We're asking you to answer again. But then there's also a lot of long form questions in there that are asking how well you understood the presentation, what might help you understand it better. And I implore you all to please answer those to the best of your ability, because that feedback is going to help us develop educational resources in the future in preparing for NF clinical trials.
Mm-hmm. Yeah. And feel free to also send questions to us through the app if you'd like to, through the rest of the summit.
Yeah, that's going to be open throughout the summit. So if you think of a question later, just come right back to the session and answer the question, and Kara and I will be monitoring it to answer.
Mm-hmm.
All right.
So thank you, everyone. We have a 10-minute break now, so appreciate your attention.
Thank you.